Okay, so let's get started. So I want to take a minute to orient ourselves where we are in this class. As you recall, we spent several lectures up front talking about biochemistry, genetics, genomics, and so forth, and how all that information can be put together formally into something we call a reconstruction. So we went through all of that material. Then we started part 2 of the class, where we converted all that knowledge into a mathematical description. And we studied the properties of that mathematical description, particularly the stoichiometric matrix. And some of that material was a little mathematical. So what we're going to do now, going on to the third part of the class, is to look at the computational models that we can now build based on this information. And as you recall, we are operating in confined solution spaces, and we are going to be looking for studying, analyzing biological functions within the governing constraints that living systems operate under. So the first lecture in this series speaks about dual causality. So I told you when we went through the singular value decomposition lecture that that was perhaps the most mathematical one in this series of lectures. This one is perhaps the most philosophical one and conceptual one, and should be kind of general interest. So what does dual causality mean? There is causation in physics and chemistry that we've been describing, but then there is something called distal causation in biology, which relates to how organisms evolve and change over time. And there's a driver behind that that we're going to talk about. And if you don't understand the history of living systems and that driver, it's hard to understand biology. And that's what that quote there at the bottom of the slide basically says. And I'm sure you've heard this quote many times before. All right, so what are we going to talk about? So I'm going to start out by talking a little bit about causality or causation and how we think about causation, both in physics and in biology. Then, you know, having outlined what the causality is we got to deal with, we have to think about how to mathematically represent it and formulate genome scale models. So we'll talk a little bit about the philosophy underlying that. And that leads us to these COBRA methods that this part of the class is focused on, these constraint-based methods, and talk about just some of the fundamentals. No mathematical details, just more conceptually. And then there's a somewhat interesting segment in the lecture here about genome scale models that we call GEMS, because they are so valuable, both from a scientific standpoint as well as a practical standpoint. And talk a little bit about their characteristics, not just the ones we're going to focus on in this class, but the broader biological features that remain to be studied. A little bit about the implementation of constraint-based methods, and this comes down to appreciating how to do successive imposition of constraints. So the more constraints you know, the more you can impose and use. And at the end, maybe give some tongue-in-cheek analogies between constraint-based thinking and our everyday life experiences. That kind of sometimes helps to put this in context. All right, so let's first talk about causality or causation, this notion of cause and effect. And this is what the natural sciences and physical sciences are really all about, try to figure out what we call the laws that describe the nature and the universe around us. How do we think about cause and effect? So let's first briefly talk about physics and chemistry. How do we think about causation there? You've all had many, many classes on subjects probably ranging from quantum mechanics to PChem, the chemical bond, all of these things. And how do we think about causation in physics and chemistry? Well, normally it's a procedure that follows somewhat these steps that I'm going to talk about. First, you identify a molecular mechanism or some fundamental process of interest to you. I don't know, could be how the nucleus in an atom works, how is it comprised, how does it function. So there's something you want to study. And you start by developing relationships based on fundamental principles. You normally have to simplify these because you can't solve them. For instance, like the Schrödinger equation, that's very fundamental, can only be solved in 4 cases, but yet in principle it applies to everything. So you normally have to do some simplifying assumptions. I should say apply some simplifying assumptions, formulate a theory, then you test it experimentally, put it to critical tests to see if it describes the phenomena accurately. And once you're done, you think you understand it. Now that brings us to this quote here, we'll see also later in the class. Nothing is more practical than a good theory. And why is that? You have a good theory, you only calculate. You can design things in silico. You can build, for instance, aircraft simulators. You don't have to train pilots in a plane because you can simulate the plane well enough, as an example. And the result of this process are equations like this that you've seen many times. Equations, the basic equation of theory of relativity, osmotic pressure, or diffusion. And there are dozens of these kind of laws that we learn about and apply. So that's how we think about causation in physics and chemistry. Fundamental principles described by pristine equations, and we can use them for all sorts of things to explain the world around us. And of course, these have proven to be foundational to a lot of engineering design. So let's just go through one example. We are all familiar with the notion of a random walk process of a molecule. Is moving in one direction, hits something, bounces off in another, does it again and again and again. So here is a computer simulation of a molecule in a gas phase bouncing around and developing this trajectory that is simulated for an individual molecule. So that's kind of the microscopic process we are interested in. And that causes an effect that we recognize macroscopically as a diffusion process, where molecules tend to move on average from a more concentrated region to a less concentrated region, which is kind of an averaging of a semi-infinite number of these individual trajectories. And that we describe by Fick's law, which is the equation there at the bottom. And in that case, we may have a probability distribution to deal with, like the one shown at the bottom, where the molecule starts at point 0 at time 0. Then we try to calculate the probability of where it is at some time later. And we get this distribution function that kind of broadens with time as it has a chance to move further away. But still, it's always most likely that it's exactly where it started, interestingly enough. That sometimes feels that way in research, right? You try and you try and you try, and you're still where you started. Okay, so causality in biology is thought about quite differently. So we, of course, in biology, we're thinking about replication of organisms, and we think about the generation of non-identical offsprings, which leads to the generation of diversity, and we talk about a biopopulation. We can talk about a population of molecules like I had on the previous slide, where all the members of the population are identical. In biology, the populations are comprised of individuals that are non-identical, and we think about that as a biopopulation. So that population is, functioning, growing, trying to do things, and some succeed and some don't, or some succeed better and others don't, as we know, and that leads to this process of natural selection. And over a long period of time, that leads to an evolution of an organism that we can study on a short time scale or a long time scale. So we're all familiar with this. This diagram here might be a little less familiar to you. And this is quite an important diagram conceptually. So what's shown here are two spaces. On top, a space of genotypes, and at the bottom, a space of phenotypes. In biology, distal causation, this process described on the left, is really an iterative loop between these spaces. So for eukaryotic organisms that undergo sexual reproduction, we can start that process in the genotypic space, where two haploid versions of a genome combine to form a diploid versions embodied in a zygote. That zygote is then projected into this phenotypic space, where an organism takes on a physical manifestation. It develops it grows, and if it can successfully reproduce, it shoots an alternative back into the genotypic space. You have another combination, and the loop goes around and around and around. So you can think of distal causation in biology as this endless loop that is constantly iterating between the space of genotypes and the space of phenotypes. And you can walk through this process. So anything that happens in that genotypic space for any organism, not just sexual reproduction amongst eukaryotes or eukaryotic organisms, there are many different processes potentially at work in that genotypic space, like just mutations upon division in prokaryote, horizontal gene transfer, gene duplication, segmental amplification. So there are many, many mechanisms at work that can change the genotype. In the phenotypic space, this iterative loop, endless iterative loop, is more constrained by physics and chemistry. How can an organism function? What environment does it find itself in, and so forth. And so there are some constraints on that, of that sort that constrain progress in the space of phenotypes. So if you want to look at this philosophically, this is an endless iterative algorithm without any obvious endpoint. So, what is the ultimate purpose of life? Where do we end up? What is the purpose of life? Just to keep this iterative loop going forever so the organisms and all their variants actually survive. But anyway, that's how you can describe distal causation mechanistically or algorithmically in a very abstract way. Now, when we think about systems biology, we have to think about both of these types of causation. And we are stuck with building models that describe dual causations, both of these at the same time. So first, living systems, when they're operating in that phenotypic space, they have to obey the laws of physics and chemistry. And when we impose them mathematically, they take the form of bounds, as shown in the first sub-bullet point there, like concentrations are non-negative variables. They can't be negative. Vmax is constrained the absolute value of fluxes, and we talked about that, thermodynamic constraints, and so forth. So a bunch of inequalities. But there's also a bunch of balances on conserved quantities, physical quantities, like mass and energy, elements, and so forth. Often you have to consider osmotic pressure balance across an unsupported membrane, because bilipid layer can really only tolerate a few millimeters mercury of normal pressure. So cells that don't have a cell wall have to be balancing osmotic pressure all the time, and that's a constraint on their function. Then, like I said, at the same time, living systems obey a genetic program with successive generations, and involved in that is willful generation of alternatives, like recombination during meiosis, as an example, and that leads to a natural selection, which really, when you look at it, is actually a mindless process, but when you look at it at a distance, it looks like it has a purpose, it looks like it has an objective, but that's just the clearing of the bad alternatives and the selection of the good alternatives. So, but still, there is some sense of optimality built into this process. Some alternatives are better than others, given the environment and some other conditions. Okay, so distal causation, sometimes called ultimate causation, results from this constant experimentation with the genomic sequence and selecting fit alternatives. Maybe not selection of the fittest, but selection of a fit alternative that survives. It's really survival. It's maybe not just survival of the fittest. And then there is the proximal causation, which is the reaction of an organism against a constant genetic background to various perturbations or changes in the environment. Okay, so to contextualize this a little bit in terms of the discipline of systems biology, we are dealing with cells at the genome scale that we've talked about, and There's a genome scale image based on a biophysical view of a cell, all the molecules in there organized, and little call out there to show the multi-scale nature of this. What we are trying to do in this class or in this field is to build this relationship between the genotype and the phenotype that we've talked about before, going from this world of chemical causation, all the PChem issues we talked about, to the world of biological causation at the same time, while accounting for these two types of causations, dual causation. And as the diagram indicates, cells may be comprised of a lot of components, thousands of them, but they don't behave independently, because then you have chaos or anarchy, which does not lead to higher order structures and an organized function. So modularization in systems biology is a big topic, as we talked about. And the min spans, for instance, we spoke about a couple of lectures ago, is one way of carrying out that modularization. So here's an example I like to give this contrast between proximal and distal causation. And this comes from the world of bioproduction. And as you know, people engineer strains highly to have a particular desirable process phenotype. And that would be mostly the production rate of a product and the growth characteristics of the organisms. You want those two. And you engineer them. And so if you start out here at the x-axis of 0, we may have some production rate of an organism designated by that point, high on the y-axis. If we let this organism go, it'll normally lose that property, because this is a phenotype we have engineered. It has nothing to do with natural selection or fitness of an organism in a natural environment, even in the fermenter that we are trying to grow it in, or the cell culture device that we have it in. So that property, this proximal causation of responding to a substrate and generating a product, will disappear over time as the organism starts evolving. And of course, the way that's dealt with in practice is that you only carry out batch processes in this field, and you have a cell bank of multiple vials of this organism that you grow up in a very systematic fashion and inoculate your process, and you do it over and over again. So you always stay kind of at x equal to 0 in this on this diagram. In contrast, we now might be able to use genome scale engineering to growth couple that phenotype. So and then we can select based on, say, growth rate pressure, selection pressure, and then that trait we are after may be improved. And this is one consequence of genome scale science, that it is possible, at least conceptually and in practice for a few demonstrated cases, is that you can couple the proximal phenotype you want to the evolutionary process that you can impose on an organism. This is kind of fascinating because if you have a self-optimizing strain in a bioprocess setting, it'll self-optimize it over time. And the only downside of a continuous process there would be contamination. Although if you have a self-optimizing strain under that condition, it may be not so likely that another organism comes in and outcompetes it, but still that would be your remaining risk here. Okay, so those are just some comments about dual causation, and here are some books, general books about this that I've about this topic or this subject that I've come across over the years that I like. So Ernst Mayr was a biology professor at Harvard, died, I don't know, half a dozen years ago or so, at the age of 100. So he was born in the early 1900s. And when he was coming of age scientifically in, say, the 1930s or so, when quantum mechanics was science and biology was, classification science, library sciences, I think Sidney Brenner called it at that time, just clustering and classifying. But he knew, of course, that wasn't the case. And, all his career, he was fascinated with this question of how is causation in biology uniquely different from that of the other natural sciences or physical sciences. And he has written books like this, which are interesting to read. Then there is the Delphic Boat, written by Antoine Danchin, who is at the Pasteur in Paris, was chair of the microbiology department there, and he wrote this remarkably interesting book in the late 90s about genomes. You know, the first genomes were out, and the subtitle there is What Do Genomes Tell Us? And it's a fascinating story. You know, there's a, well, I should back up, there's a fascinating story in there about the achievement of sequencing the first microbial genomes. The title of the book comes from a question asked by the Oracle of Delphi of the mortals, and it goes as follows. Imagine you have a boat, like the one shown there, which at the time was made of planks. If you own it over a long time, planks will break or rot and you replace them. And given enough time, every single piece of wood will be replaced in that boat, which gives rise to the question that she asked, is it still the same boat? I mean, is the design, the whole, more important than the parts? And of course, this is a fundamental question in systems biology. And I liked it so much that when I was lucky enough to be able to buy a boat, I called it the Oracle. So that's my Delphic boat. Then there's another book written by Richard Feynman, which is also interesting, very short book. As I think we all know, very famous physicist operating at Caltech in the 60s. He wrote this book called The Character of Physical Law. So he goes through the characteristics of all these equations and kind of from a more broader, almost philosophical perspective. And it's interesting to read that to get a better understanding of what causation is really like in physics and chemistry. So those are some interesting, you know, relatively easy to read books that you might consider to pick up. All right, so now, against this background, we're going to have to ask ourselves, how do we do mathematical formulation? If you go back to the first slide on, say, or the second slide on diffusion, we know approximately how we're going to do it. You do some statistical assembly, some averaging, you end up with differential equations that describe the phenomena very precisely. But how do we do it here, knowing how sloppy biology is and how it produces multiple outcomes? For instance, everybody in this room is an equivalent solution to a selection process. We're all here, we're all surviving, but we're all different. That doesn't exist so much in physics. There's the solution. But in biology, there are many solutions. So for many years, people have tried to build rigorous biophysical models of biology and not succeeded. And there are some fundamental and practical reasons why. The first one is, and recall that image that I had up a minute ago of E. coli, the intracellular environment is very complicated. Thousands of different molecules, different abundances, and so forth. If we want to take the approach of physics, how are we going to define the geometry? Well, it's going to be changing. How are we going to define initial conditions? How are we going to define boundary conditions? How are we going to write out the equations? I mean, just how do we start by just formulating the problem before applying any assumption? Is it a continuum in there, or do you need to be counting molecules? What is it really like inside cells? I personally think that in time we will view cells as the fourth state of matter. It is not a crystal, it's not a liquid, it's somewhere in between, it's highly organized, there are long-range forces that operate across a cell, electrochemical and likewise, and there's something very different about the physics of the intracellular environment. And I have many examples I often like to talk about in this regard, but we won't do that today. So the second issue here is a practical one. First one is, I guess, fundamental. The second one is a practical one. Let's suppose you could write out all these equations, and you ended up with, say, 100,000 equations to describe an E. coli cell. Equations like that can be solved, actually, when you design a new chemical plant, you know, by the coast of the Gulf of Mexico. That's what chemical engineers do. They solve these kind of equations that describe, you know, chemistry reactions, osmotic pressure flows, and stuff like that. But where are you going to get the parameters from? I mean, we know already that in vitro derived parameters based on something you isolate from a cell does not represent typically what happens inside the cell, and we can't measure a lot of parameters in an intact cell. So where are you going to get the parameters from? That is probably an insurmountable issue. at least if you wanted, to describe every little detail of a cell. Of course, you can coarse grain and maybe make approximations, but in principle, these parameters are very, very hard, I think impossible to get. But let's suppose you even could. You have this E. coli clone or yeast clone or whatever, you get all these parameters. Now you start applying it. Well, what's going to happen to you now? Well, the cell is going to diversify right away based on uneven divisions and diversification of alternatives. So even if you have a pristine parameter set, after a few generations it's lost. And if you look again around in the room here, we all share the same genome, more or less, but there are sequence variations in it. So we all have our own individual parameters. So I can get my parameters for, I don't know, some function in the liver, but it wouldn't apply necessarily to you. And that's kind of the basis for personalized medicine, as we've talked about, I think, earlier. And I guess it's now called precision medicine. But these are both fundamental and practical reasons why it is very hard to take, I think actually impossible, to take the same approach to model building in biology as is taken in physics and chemistry. So what are the alternatives for genome scale models? So the alternative that has emerged is this constraint-based modeling. That's a very practical one. It's data-based, it's based on fundamentals of physics, and it can predict a number of biological functions, as we talk about in the fourth part of the class. How broad that spectrum of predictions of phenotypic states is yet to be determined, but it seems to be broadening by the day. So the third limitation here basically results from dual causality, this uneven parameter sets, say, between individuals in a biopopulation. So one way of dealing with it, or an alternative to building rigorous biophysical models, is to do this genome scale reconstruction process that we've talked about, identify all the constraints that we can find that limit the functions of that network, define inputs and outputs and some other model parameters we talk about that are relatively few and can be experimentally determined. That gives us a set of solutions, but not just anything. It gives us these solution spaces that we talked about. And then we actually try to guess at what is the most likely biological functions under those constraints. And that calls for an optimization process, or more generally speaking, a constraint-based optimization problem. So that's a procedure that works, but what emerges from this approach is the objective function. And it used to be felt that there was the limitation of constraint-based modeling, but over the last 10, 15 years, it's become clear that the objective function is the function that describes distal causation. It's the biologist's function. It doesn't exist in physics and chemistry. It's a biology-based function. And we can now study it by laboratory adaptive evolution as an example. And we have a whole lecture on the objective function coming up because it's very philosophical. It's also very practical. And this is what makes constraints-based modeling so interesting, actually, fundamentally. Because it deals with, some people may say purpose in life. Others may call it a success criteria in a selection process. So here is the philosophy of constraints-based analysis described by a quote to Sherlock Holmes, where he says, how often, says to Dr. Watson, how often have I said to you that when you have eliminated the impossible, whatever remains, however improbable must be the truth. Now this really describes constraints-based modeling very well. It is the quote I put at the beginning of the this part in the book. And this quote was actually suggested by Marcus Covert, who is now a professor at Stanford, graduated here in 2004. And the panel there to the right kind of describes the overall process. We reconstruct, we impose the constraints, we form that solution space, and then we study it. Simple process, right? 3 steps. But the implementation It may not be quite that simple. As I was preparing for this lecture, I actually came across this photograph that I thought I should show you. So here's Marcus Covert in the blue shirt there. And that is Sidney Brenner that came to visit us here. This is maybe 3 years ago, 3 years ago or so. And this picture is actually taken right outside the lecture room. Okay, so let's talk then a little bit about the constraints approach to describing biological function at the genome scale. This is what we're talking about, the genome scale. So here's a very early, I think the earliest conception of the constraints-based approach to studying biological functions. This paper here realized that there are outer constraints on biological functions simply based on network topology, like the stoichiometric matrix and what it represents. When you map the kinetics on top of that to get you mass action kinetics for the individual reactions, you shrink that solution space, that outer one that is blue to maybe that one that is green, which means that you now have to get the kinetic parameters. And that space may or may not be convex. It could be concave. And when you build sometimes these kinetic models, you come up with multiple steady states, unstable regions, dynamically unstable regions in the space where you can't have any operations. You could even segment the space and so forth. So very complicated, actually, mathematics, and requires the parameters that I spoke of earlier. So you can study maybe the natural mass action tendencies of a network you put together, and you can study, you know, the numerical values of the kinetic parameters and do that. Then you can impose on top of that the willful regulatory mechanisms that are so prevalent in cells that bias the natural stoichiometric mass action-based tendency of a network. That's what regulation does. It moves it away from what the chemistry really wants to do by building regulatory mechanisms. And then finally, because, like I talked about, the kinetic constants can change with evolution, the shape of that green space can morph and change, and it can move around in that outer space, but they got to stay within it. So at least if we can define the outer space and state these optimality criteria, we may have a shot at understanding what that green space is like and how it may move around if we evolve an organism and so forth. So then some more specific statements started being made about these constraints. So here's a table that outlines some of them, and there are many more of them we'll talk about later. We have a whole lecture, in fact, coming up on constraints. So this is an introduction to them. So there are physical chemical constraints, like I talked about, the osmotic pressure, electron neutrality, solvent capacity, molecular diffusion. And these are basic physical laws that cells have to abide by. And these are hard constraints. These are non-negotiable. These cannot be changed. So you can use these to identify that outer solution space that I talked about. There is connectivity. I mean, once you define the set of reactions that take place inside a cell, you have the topological properties of the network. These are also hard constraints, unless there is a, say, horizontal gene transfer event that takes place. You acquire a new enzyme, and all of a sudden there's a new link now in your network. So the topology can change with evolution. You can have segmental duplication of the same genes, and then asymmetric evolution of them. And so one link may become two by that process too. It doesn't have to be horizontal gene transfer. But if you can define the stoichiometry, the network connectivity in a particular organism, these represent non-negotiable constraints. And presumably, say, for us in the room, we share the same stoichiometric matrix for our metabolic network, as an example. Whoops. Then there are the capacity constraints that we also talked about, that there are maximum and minimum values on all variables in the physical world, and we can state these and post those. And these tend to be non-adjustable, at least in the outer sense. So a Vmax may be absolute at the highest protein concentration achieved, as an example, but you can throttle this one down. So you can increase that constraint, but you may not be able to alleviate it. So that's maybe a one-way adjustable constraint. And then there are the rates, the reaction rates. Those are highly malleable. And those are all subject to changes in the DNA sequence. And in fact, when you start evolving cells in the lab, resequencing, you will find the mutations in regulators that wobble maybe the composition of the cell, and you will find mutations that change the kinetic properties of the components. So this is where a lot of the action is, at least for short-term adaptation. Okay, so these are some comments about constraints. So let's now talk about the COBRA methods and some of the fundamentals that come with them. So early on, I listed some axioms that underlie the COBRA approach, and here I'm going to restate them just a little differently. The first one is that cellular functions are based on chemistry, and I think that's an absolute statement that we won't argue with. And these can be gleaned from genome sequences and from the literature, and we can assemble all that chemical information. Cells, they operate under a series of constraints, and we talked about some of them. Many of them derive from the physical laws, like mass and energy balances. Cells function in a context-specific manner. We've changed the environment from one place to another. They know that, they respond to that, and they react to that through regulation mostly. And we can now study that by polyomic data profiling, just collect a lot of different omics data types in a given environment, and now you can map that onto a network as constraints and do studies with that. And then, of course, cells evolve under a selection pressure in a given environment, and that is the basis for the iterative process that I talked about before that leads to the phenomena of distal causation. Okay, so that's what underlies the approach. Now, as we've seen, when you start applying it at the genome scale, you end up with a genome scale model. We're going to be studying them, these gems. So what I want to do now is not so much talk about the mathematically in detail, but some of their properties, if you just look at them qualitatively. So let's first talk about the generations of gems. And they are developing successively based on how many constraints we are putting in into them to define them. So the first generation of these constraint-based models are based on genomic data and bibliomic data, and they define that outer solution space. And they can help you understand what is possible for an organism to do. They can answer that question, or more importantly, maybe what is not possible for an organism to do. Can kind of divide, distinguish between these two possibilities. The second generation of these models would include transcriptomic and proteomic data, where you just map them in a condition-dependent manner. And that tells you actually what states were chosen by whatever regulatory mechanisms were there. And this has been done, for instance, for human tissues. You can map proteomic data or transcriptomic data onto Recon 1 or Recon 2. And you can customize the network for that cell. You contextualize the network for that cell based on its composition. So there are a few of these models available, second generation models. And there are some third generation models becoming into being now. And they are based on the fact that we can get a time-dependent set of these omics data set. Not just two different states and two different, under two different conditions, but a time series of omics data sets that help us to figure out how the different states are chosen. And so there are workflows beginning to develop for these alternatives. So these are the generations of them, at least as of now. Maybe there will be a fourth and a fifth generation, who knows. So let's talk about some of the features. What are the features of GEMs that distinguish them from models in physics and chemistry? The first one is redundancy. There's a lot of redundancy in biological systems. And we see alternative optimal solutions. So we're going to talk quite a bit about this concept and phenomena later on. They're sometimes called silent phenotypes. And icon A talks about that in the image there. It shows two isozymes that catalyze the same reaction. And the first one may have been the predominant one, and if you knock it out, the other one may take its place. And so there's a molecular phenotype because you switch between enzymes, but you don't see it in the function of the organism. And it's called a silent phenotype. Or this may also be called non-causality with respect to between the genetics and the phenotype. So you see this everywhere. You see this in the genetic code. One tRNA can translate multiple codons. One codon can be translated by multiple tRNAs. You start looking at this. This is a very prevalent property. Very few critical points in the biology. Amgen was lucky enough to find EPO. That is one of them. So if you can find something like that, that will be quite important. The other is multifunctionality. And that's shown by, for instance, in that middle of the panel by a multifunctional enzyme. One enzyme shows up, does many things. One tRNA translates many codons. So this leads to a certain notion of a generalist phenotype, and we'll talk about that later. But multifunctionality is seen broadly in biology. And then there's one I won't talk so much about, but there is causality and there is non-causality in biology. So remember, we have hierarchical layers in this description between the genotype and the phenotype. One example of non-causality is a hitchhiker mutation. And I think you've heard about those. So there is one causal mutation that is being selected for, and then there's another one just selected for at the same time. It may be in the wobble base pair position in the gene and won't change the amino acid. It won't do anything, but it's selected for, and it shows up, but there's no causation from it. So you often see a lack of causality between the hierarchical layers. And of course, that's important if you want to develop any multi-scale theory in biology, because you need to understand the events that actually propagate up that hierarchy and those that don't. Gödel's theorem, of course, always leads to a long philosophical discussion that we're not going to go through, but truth is a stronger property than proof. You cannot prove all truths. You can't be complete and consistent at the same time. So genome scale models want to be complete, but maybe they can't be consistent. This is, of course, the politician's dilemma, to be consistent in everything they say and please everyone. Not possible. But fundamentally, genome scale models, I think, in the end, will be interpreted in terms of information flow. And I didn't want to put Shannon's law down there or anything like that, because it may not be the sort of an information theory that we know today. But there's some way of describing information that will ultimately find its way into genome scale models. So along those lines, let me say a few more things. So this becomes a little more almost philosophical. If you have a true genome scale model that describes the relationship between genotype and a phenotype, you should be able to do a number of things. So let's talk about that here. So there are some higher order functions in biology, aside from rate constants, binding constants, and things like that. And they include a few features we should observe. One of them is self-assembly. We know that when you put a bunch of components together from a single cell, they may self-assemble. They know that they fit together, and they just self-assemble. Nobody forces them to do it, it's just built into them. So presumably this property may result from having structural biology put into the models, so we can see how the pieces fit together, and maybe what are the thermodynamic forces that force that assembly to take place. Selection is definitely at work in distal causation, but there's more and more evidence that even proximal causation, as this hierarchical system is trying to respond to something, also acts by selection. And this hit me at least first when I saw some experiments out of Irv Weissman's lab at Stanford, who was a pioneer in stem cell biology. And this was back in the late 80s when he was studying hematopoietic stem cells in mice. So briefly, hematopoietic stem cells become progenitor cells that are lineage specific, and then they generate the progeny, say the red cells or the platelets or whatever. He showed that only about 1 in 1,000 progenitor cells generates all the progeny. What happens to the other 999? There must be a selection process of some sort that decided only 1 in 1,000 was fit to do the whole work. that was needed to generate all the cells. So I think when we start even looking at genome scale models in terms of proximal causation, we will be thinking about selection. Adam Arkin at Berkeley argues this is the same case based on just statistical properties of molecular events and how reaction happens. And there is actually a selection process built into just the random thermal motion of molecules. So it may be at all levels. The notion of self in biology is very important and fascinating. I mean, you express a foreign protein inside the cell. Ooh, it knows it doesn't belong there. It may misfold, it may activate a protease and get rid of it. So this notion of what is self in biology has to eventually find its way into a genome scale model, and it may be related to that first property. A protein that is heterologous goes into a cell. It won't self-assemble with the other components, and it therefore, is not a part of the group. It's an outsider. And I think there's another notion that's quite important. I think awareness is a fundamental property of genome scale models. Cells are extremely aware of their environment, and they behave accordingly. And the functional states that we are trying to calculate for networks really reflect awareness. Because the cells, in this context-specific manner, they know where they are, they know what to do, and they're aware, and they do what they need to do. So there's some awareness is built into these models in an abstract way. So maybe for future generations of practitioners in the field, we can maybe take these fundamental biological concepts and define them mathematically. Wouldn't that be interesting? I mean, that would really raise the profile of molecular systems biology that was possible. We already in the lecture on pathways saw parallelism between biological and mathematical concepts. Pathway is now a basis vector in a space. I mean, you would not have imagined that 10, 15 years ago, that a pathway actually has a rigorous mathematical definition when it comes to genome scale science. Who is to say that the same thing won't happen with these properties? I told you this might get a little philosophical. Okay, so maybe back to practical things. How do we implement constraints-based thinking? So first we have to define constraints and figure out how to impose them. So here is a classification of constraints into 4 categories. The first one we talked about a lot, physical-chemical, that comes down to applying the laws of physics and chemistry. There are the topological constraints we've also talked about. So there are topological properties of networks, but there are also topological property in terms of biophysics. The genome, for instance, is wound up in a certain way in space, and we don't quite understand what the function of that is, but there are constraints associated with that. The inner membrane in the mitochondria folds in a very particular way, and those represent topological constraints as well. These two, the physical topological constraints are by and large not accounted for yet in genome scale models. You can take the membrane area maybe and say the maximum protein loading in a membrane is 40% of the membrane and put that as a constraint. You can take the fact that 15% of the wet weight of a cell, or about 55% of the dry weight is protein, and say that's the total number of protein you can have. So there are rudimentary overall constraints like that can be placed on models, but that needs to be worked on in the coming years. Of course, the environmental constraints, those are relatively easy to measure and define. And then there is the notion of regulatory constraints. And these are fascinating because these are self-imposed. This is almost like an internal selection process inside the cell to pick the best set of molecules to be expressed in a given condition. We'll talk more about that in a moment, but that really is no longer a constraint, it's a restraint, it's self-imposed. And this is where a lot of the selection process, where the selection process is reflected, and how a cell biologically walks around in that outer solution space that I talked about earlier. So you can use different classifications of these constraints, and there are examples given in the textbook about how some leading biologists have thought about this over the years and how they have stated them. They basically come down to the same classes of constraints. This is maybe an operational classification because we can group them in this way and impose them onto genome scale models. Okay, so how do we use constraints? Often they are imposed successively, so there's like successive imposition of constraints. And this is an old image that shows that. And the first one starts with an unconstrained space. And the first constraint that's imposed here is the dynamic mass balances, and that leads to a plane, that null space that's a plane. Then you may impose the non-negativity constraints of the fluxes, and now you get a cone that is convex. Then you may impose the maximum constraints, and that closes off the cone, and now you have a closed solution space. And then there is a selective group of kinetic constants you can actually impose that'll segment that space, and it'll build from it a union of convex space that in their totality may not be a convex space. So that's the idea of using constraints, is to successively impose them. And of course, you really always want to impose the most dominant constraints. It doesn't make sense to be imposing redundant constraints because that doesn't do anything for you. All right, so at the end here, I want to speak to this a little bit broadly. So the first thing is the analogy to engineering design. Many of you are educated as engineers, and this will resonate with you. And this was actually inspired by my thesis advisor at Lightfoot when he was first exposed to this way of thinking some 15 years ago. He said this is just like engineering design. So what is engineering design like? So here's how it goes. If you have to design something as an engineer, well, you have an objective. I want to separate all the protein in E. coli as an example. Or like a structural engineering department here at UCSD, they want to build a composite bridge across I-5. They've been talking about that for 20 years and haven't done it yet, but that's what you want to do. So there is always an objective that you design against. And the first thing you normally do when you build a design, you look at your constraints. Geometry, that bridge has a certain length. You know, that's geometrical constraints. What materials I can choose from. What are the physical chemical properties that I have to deal with. But most of the time, the governing constraints are cost and time. This is your budget, this is the time you got. And what you do with these constraints is you define what's called the design envelope. And that is fixed by some of the parameters, but then you identify what are called free parameters, and they can vary, and you can optimize your design based on those free parameters. And I like to point out that optimal engineering designs do evolve. And as some of you know, I used to be a professor at the University of Michigan, close to Detroit. And there is a museum of industrial history there. And you can go there and look at the industrial designs, like the first sewing machine to the modern sewing machine, the first dishwasher to the modern dishwasher, trains, cars, and so forth. And you can just see in front of you how engineering designs evolve. And they tend to evolve, I think, based on availability of material and human desires for style and convenience, probably. So if I put this up side by side with a constraints-based modeling approach, you see the analogy. So we have a design objective. Cells also have an objective, fundamentally to survive, maybe to grow fast, maybe a Helicobacter pylori to survive the low pH in the stomach. You have these objectives. Normally we don't know them, which is interesting. You can maybe guess them from the evolutionary history of the organism. But now we can prospectively define them in experimental evolution settings. So you can actually study objective functions now experimentally, which is so exciting. You state your constraints, like we talked about. One side, you define a design envelope. We call it a solution space. And then for engineers, they optimize using the free design variables. And like I pointed out, for biology, at least in the short term, those design variables are kinetic constants and may be regulatory phenomena. That's the quickest way to change your design. And since regulation is so important in this process, and I mentioned it earlier as restraints to find a portion of the solution space you like, we talk about that now as, broadly speaking, as a bi-level challenge to cells. The first is to decide which gene should be expressed, and that's the issue of transcriptional regulation. That'll shrink the solution space for you. Either we have the regulatory rules when we build the TRN, or you can simply measure which proteins are there or which transcripts are there. And with that, there are a number of methods we'll talk about. You can shrink the solution space. But it doesn't go down to a point. It goes to a smaller solution space. And then regulation of the activity of the gene products will then take the second step and locate you more precisely in that phenotypic space. And this is kind of where you might think about it. So this pentagon, kind of a reflection of that little image I started out with, may be your outer solution space. And if your best biological function is at the apex of it, at the top, represented by the green dot, all you got to do is to shut down the rest of the solution space and make sure that that operation is never carried out. You don't have to know where the red dot is. You just have to know where the bad alternatives are. And this is why I think selection pressure or selection-based process may even work in the proximal causation, just the way a cell responds. It really doesn't know exactly what to do, but they know it can figure out what works better than other things, and it kind of shuts down the bad alternatives. Okay, and there's still a gray zone there, so it's not going to be completely collapsed to a point. And this leads us to what I talked about earlier. Sampling can help us determine the dimensionality of these two problems. You can look at the dimensionality of the TRN, what are the effective dimensions there, and then you can look at the pathways that tend to be upregulated in a certain point and determine the major pathways that do need to be regulated in terms of enzyme activity, as an example. So that's how, in constraint-based modeling, you can break this problem down into two separate ones. And as you know, they operate on two different time scales. Metabolic responses are quick, changing the proteome composition in a cell is slow. So another hierarchy for you. Okay, so let me now talk a little bit about the different point of view you start to adopt when you look at biology after adopting the constraint-based thinking. So let me just give you two examples. So let's talk about the process of oncogenesis, the formation of a tumor. So this image here is obtained from that review cited at the bottom, and it divides the oncogenic process into about 7 steps. And each one of them is really the removal of a constraint. So think about that pentagon. You have all these constraints to get the physiological function you want. If any of these constraints are lost, you can wander into other portions of that solution space and take on different phenotypic states. And what this diagram shows is how that process is now subdivided into 7 distinct sets. And there are the pathways involved in them, there are the phenotypic consequences of that. And so this is a successive alleviation of constraints to get a full-blown metastatic cancer. And what's interesting when you look through that list of the mechanisms that underlie these, what are called here carcinogenic pathways, there are alternatives. There are different ways of overcoming these constraints. So there are different optimal solutions, well, optimal from the cancer standpoint, to get to a fully fledged metastatic state. So you can think of cancer now like this. You can look at other processes of interest to you. For instance, salmonella infection. So salmonella has to overcome many constraints to successfully infect the host. And some of them are illustrated here. It's get into the host, it has to survive going through the stomach, it has to adhere to the lining of the gut, it has to cross the epithelial layer. If engulfed by a macrophage, it has to avoid being digested by ROS species and so forth. And interestingly enough, an organism like Salmonella has pathogenicity islands, which are called spy islands, that it upregulates to overcome each one of these constraints. So if you now want to study the infection process, it really comes down to figuring out how an organism overcomes these constraints in attaining its optimal objective, which is to infect the host. Okay, so we have a few more minutes to go, so let's maybe move into some more tongue-in-cheek examples of this. So everybody knows this game. So this is a game of constraints, right? So we have a square there of 9 by 9, well, 9 rows and 9 columns, and it's divided into 3 by 3 cells. And what you have to do is you have to place the digits 1 through 9 in every one of these 3 by 3 cells in such a way that each digit only appears once in every row and every column. So you have a map and you have constraints. And now here's one example of how you figure out an answer by imposing the constraints. You have to place the number 5 in that top right 3 by 3 block, and you can just look at the 5s in the other rows and columns and figure out that 5 has to go into that green spot. So this is how you, in this case, you get a unique answer. I think there's always unique answers to these puzzles, maybe there are not. But you get an answer based on constraint-based thinking. So here's another one you might have heard after the Super Bowl. This is what you hear very often on TV from a supermodel or a rock star or a star athlete. You have one life to live, and you have to make the most of it. So the constraint, one life, and the objective, to make the most of it. Not very well stated here, the objective, but nevertheless, an objective. Here is one to Winston Churchill. You can trust the Americans to do the right thing after they tried everything else. So eliminate the bad choices, get the good one in the end. And you can go on and on. You look at this around you every day, you see this. Okay, so let's summarize here. So like I said, this is a somewhat philosophical and conceptual lecture, but it kind of sets the stage for the constraints-based approach to developing genome scale models. So systems biology really strives to bridge that multi-scale relationship between the genotype and the phenotype. So if you want to build models, quantitative models of that multi-scale relationship, we have to look at causation. I mean, to look at the causation, we have dual causation to deal with, both the laws of physics and chemistry, and then the biological causation. So we have to deal with those. We also know that all biological functions are subject to myriad constraints. And it's important to note that they are normally most dominant at the lower levels of the hierarchy, and they permeate up the hierarchy. So a constraint set at the lowest level cannot be violated at the highest level. But we also know that some of these constraints here may have causation that doesn't permeate either. So there are non-causal events like we talked about, like a hitchhiker mutation. So the constraints-based modeling formalism can account for all these considerations, all these requirements that we are putting on building genome scale models. Definition and imposition of constraints becomes a key issue. And so does the statement of the objective function, like I mentioned. And one thing that is worth noting here at the end that results from these considerations is that you view regulation as willful imposition of constraints to pick good alternatives given all the other constraints. And so they may be viewed as restraints. Okay, so that is the end of the introduction to constraints-based methods, and I should say it's philosophical and conceptual foundations.